Hello, and welcome back to Mammalian Anatomy at Keuka College. Today we're going to continue our conversation about Chapter 13, the central nervous system. And in this video, we're going to focus on the development of the brain. So how does the brain form in utero? So the brain is such an amazing organ. It is so complex, yet so organized, and it's just so amazing and cool. And just some fun facts about the brain. The brain has about 85 billion neurons with many, many more neuroglia. And what's awesome about this brain is that these neurons are going to synapse together in highly complex ways to integrate sensory information from our memory, and be able to create a coordinated response to our environment. And because of these complex neural integrations and these pathways, we're able to do things such as control our heart rate, be able to do daily tasks, ride a bike, talk, and even it helps develop our personality and our intellect. So the brain is such an amazing organ. But to really understand the brain, we need to understand how it develops in utero and how the basic body plan of the brain really reflects its embryonic development. So let's get into it together. So we're going to be using two diagrams from our textbook. This one will look at the development of the brain between about day 20 to about week 5. And it will look at how the neurotube will expand to form ventricles and how these ventricles are going to eventually be associated with structures in the adult brain. And then we're also going to look at this diagram. This diagram will follow brain development from week 5 to birth, and it will focus on the folding that occurs within the brain during brain development. So let's go over the stages of brain development. So the first stage will be the neurotube, and your textbook gives this diagram, and it is the most helpful diagram to explain things. Don't you agree? Notice the sarcasm. So what I wanted to do is kind of go over the development of the neurotube, because the neurotube is going to be the most rudimentary structure that will eventually become the brain. So just a little bit about embryonic development. Early on, around day 20 of embryonic development, our body plan is pretty much a ball of cells that is composed of 3 layers and a hollow space. The outer layer is known as the ectoderm, and the ectoderm is responsible for eventually becoming the skin, the nervous system, and also the eyes. The middle layer is called the mesoderm, and the mesoderm is responsible for becoming muscles and bone. And the inner layer is going to be the endoderm. The endoderm is going to wrap around a hollow space, and that endoderm will become our digestive system, and it will also become our visceral organs. So we have 3 layers, the ectoderm, mesoderm, and endoderm in early development. The brain and spinal cord are going to actually develop from the ectoderm. And what we'll see here is early on, before day 20, what will happen is a portion of the ectoderm will differentiate, so the cells will change their function, and they will become the neuroectoderm. This neuroectoderm will thicken to become the neural plate, And there will be a special set of cells called the neuroplate border cells, and we'll see that they will eventually become part of the peripheral nervous system. Oops. We'll get to that diagram later. Around day 19, so this is specifically day 19, what will happen is the neural plate will actually bend and come together. And eventually what we'll have is we'll have the development of the neural crest. The neural plate border will form the neural crest. And eventually these cells will fuse together around day 23 of gestation to form the neural tube. This will become the central nervous system. It'll form the neural crest. That's where these neural plate border cells came from. They will become components of the peripheral nervous system. And then over here, the ectoderm will fuse back together, forming things such as the skin and those sorts of structures. Why is this kind of thing important to understand? It's important to understand because this will show us kind of the basic layout of or basic structure of the brain. And when we get to the brain itself, we'll see that the brain is a collection of cells that surrounds a hollow space. This hollow space in the brain will become the ventricles that will be filled with cerebral spinal fluid. And in the spinal cord, it will become the central canal. That slide really wants to be up. And then the cells surrounding that space will become the brain and spinal cord itself. All right. So let's go into what happens to this neural tube after the first few days of development. Oh, if you wanted to have a different diagram to see what the kind of body plan of the early blastocyst is, you can see it here. We're just essentially a ball of cells composed of an ectoderm, a mesoderm, and endoderm. And here you can get a little bit of a better idea of the formation of the neural plate and neural tube over here. So if this kind of view helps, don't be shy to check it out. So we've developed the neural tube, which is our basic body plan. What happens next? Well, we're going to start to form the primary brain ventricles. So certain regions of this neural tube are going to expand and kind of balloon, and they will form the initial or primary ventricles, those hollow spaces inside the brain. And there are going to be three parts: the prosencephalon, mesencephalon, and rhombencephalon. The prosencephalon will form the forebrain. The mesencephalon will form the midbrain, and the rhombencephalon will form the hindbrain. The most caudal part, the part that's closest to our tailbone, and we'll talk about caudal and rostral in a little bit, will form the spinal cord. So the part that doesn't balloon will form the spinal cord. And this development, this formation of the primary brain ventricles is going to occur at 4 weeks. During week 5, what's going to happen is we're going to develop the secondary brain ventricles. So the prosencephalon is going to develop into the telencephalon, which will eventually become the cerebrum. And it will also become the diencephalon, which will stay as the diencephalon. These little projections here will actually become part of the retina, so they will help form the eyes. The mesencephalon will not split, so it will stay the way that it is. And the rhombencephalon will split into the metencephalon and the myelencephalon. And again, still the part that hasn't ballooned or hasn't expanded is going to stay as the spinal cord. So getting back to this diagram, you can see kind of these individual structures will eventually develop into. The telencephalon will become the cerebrum, which is the very wavy part of our brain that we think of when we think of the brain. The diencephalon is going to be a deep structure inside the brain. And the diencephalon will contain things such as the thalamus, hypothalamus, and epithalamus. We'll learn more about these in a later video. It will also become part of the retina. So this little part will extend into the eyes and form the retina of our eyes so that we are capable of seeing. The mesencephalon will become part of the midbrain. Metencephalon will become part of the pons and cerebellum. And the myelencephalon will become part of medulla oblongata. In future videos, we're going to actually break down each of these structures and learn about them in a little bit more detail. So the secondary brain vesicles will occur during week 5 of gestation. Also what occurs at week 5 of gestation is the midbrain and cervical flexures. So the brain has to accommodate for space. There's limited space in the skull. And also human brains have to accommodate for the fact that we walk upright rather than walking on all fours like other animals. So our brain actually has to fold to accommodate those two things. And one of the first types of folds that occur is going to be the midbrain flexures and the cervical flexure. Midbrain flexure is going to occur around the mesencephalon, while the cervical flexure is going to occur along the myencephalon. And it's going to help bring the diencephalon and telencephalon closer to the brain stem. So that's going to be the first level of folding. Folding will continue as the brain grows. And around week 3, what will happen is the telencephalon will start to grow and form the cerebral hemispheres of the brain. So the two top sides of the brain. And this will grow in a posterior and lateral direction. And as it does, it will actually follow the shape of the skull and loop around. So it'll grow outwards. The cells will move more posteriorly. And then what will happen is as it goes towards the back of the eventually forming skull, it will rotate and come back around. So our brains have a very interesting horseshoe shape to it. This area will actually become part of the temporal lobe that will kind of loop back around. Also, as it's doing this and kind of growing and trying to fit into the newly forming skull, it's going to enclose the diencephalon. So the diencephalon will be located deep between the cerebral hemispheres. As the brain continues to grow, so we can see here, like at week 26, the cerebrum will actually start to fold and crease. So you can see the structures over here and here. Any fold that is kind of shallow in the brain is called a sulcus or sulci, to be plural. And a kind of mountain shape on the brain is called a gyrus or gyri, if we're talking about plural. And this folding is really important because it allows our brain to fit in our skull, and it also increases the surface area of our brain. Specifically, it's going to increase the surface area of the gray matter, where we learned is going to contain a lot of synapses and cell bodies. So it will increase the processing power of our brain. Not all organisms have such highly folded brains like humans do. Organisms such as mice and koalas actually have a smooth brain. So this is an interesting structure that human brains have. The development of the brain is going to continue to birth, and at birth, the brain is going to look very similar to what it would look like in adulthood. It's going to have the major regions, and it's going to have the major folds that are necessary for the brain to function. But note that the brain doesn't stop developing at birth. It's going to develop quite some time into adulthood. For example, the prefrontal cortex, which is right in front over here, is not going to develop until like age 22 to 24. And that area is responsible for judgment, processing emotions, those sorts of things. This is one of the reasons why teenagers are more apt to risky behaviors because their brains aren't fully developed to make those sound judgments yet. So, yeah, patience. Yes, no, maybe so. All right. So that goes over the different stages of embryonic development for the brain. In the next section, we're going to look at a general overview of the brain and identify the major features and what vocabulary we use to look at different directions for the brain. And in the following videos, we'll take each section and really break it down and look at the structure and function of each component of the brain. I'll see you in the next videos. Have a wonderful day and be good to yourselves. Bye-bye.